Why does the Y chromosome exist? This seems like a really inane question to ask because in our popular imagination, we think about the Y chromosome and the XY system to be as fundamental to male biology as, for example, testosterone is. But this then begs the question, why is sex not a phenotype that exists in gradations similar to, for example, height or skin color? Why are there two separate chromosomal structures that control this kind of discrete assortment of phenotype into male and female? Well, let's start with a timeline to explore why this is the case. Let's begin the story 300 million years ago with the common ancestor of mammals and reptiles, existing somewhere in the continent of Pangaea around the Permian era, which was one of the worst eras for life on Earth actually to exist, with surface temperatures reaching boiling levels. But that's a topic for another day. Now, this common ancestor, it had a separate sex chromosomal system, of course, one that we can't really determine now because it's been so long. But it had two homologous chromosomal pairs, both of which gave rise to our X and Y system. And at first, these homologous pairs were entirely identical, similar to any other chromosomal pair that we have in our genomes that are autosomal pairs. But 300 million years ago, this common ancestor started to diverge into mammals up here and reptiles. And in mammals, what happened is that one of these chromosomes suddenly gained a sex-determining region, which in mammals we call the sex-determining region Y. And the creation of these sex-determining regions is actually the focal point of speciation. It's actually why speciation even happens, actually. So very, very important maneuver in evolutionary history. And reptiles, they have their own sex chromosomal system, similar to, as they derived into dinosaurs and birds, they have the Z and W chromosomal system, which is also a topic for another day. So then let's continue on with our timeline. Mammals then eventually, started to diverge into the hominid and the primate line. And we start at the common ancestor of hominids and non-human primates. And just this is a simplified story, but what is happening here is that chromosome in that homologous pair that contains the sex-determining region Y, the one that's important for male development, well, that has to, by nature, it has to avoid recombination with the female or the baseline, the de facto chromosome, which we'll just call the proto-X chromosome right here. And that's because it has to preserve its sex-determining region, because that's ultimately how male evolution or male development arises. However, just to kind of maintain physical proximity with its pseudo-homologue during recombination to avoid being physically lost or physically misallocated into daughter cells, it has a pseudo-autosomal region at its very ends, which kind of allow for physical coupling during meiosis, but don't actually allow for any kind of genetic recombination. And along this process, every other region that wasn't the sex-determining region or wasn't the pseudo-autosomal region, they get trashed or discarded because they're not important anymore to the function of this proto-Y chromosome. And now we eventually come to us, humans, where we diverge from our great ape ancestors, chimpanzees and gorillas. We now have a structure where we have X chromosomes, of course, which retain most of their functionality. But then we have our Y chromosomes, which almost have nothing on them except for that sex-determining region, very important for male development, and the pseudo-autosomal regions, which again, I'll just allow for physical coupling of that X chromosome to the Y chromosome during during cell division to avoid mislocation. And so the X chromosome still retains a lot of its functionality because it doesn't have a sex-determining region on it. It is the baseline or the de facto, the tabula rasa. And so it still has a lot of its genetic baggage, fortunately for us. The Y chromosome has only 45 to 60 protein coding genes as compared to 1,000 on the X chromosome. And as any astute student of medicine would note, there are many X-linked recessive disorders that really point out to us how critical the X chromosome is outside of just determining sex. There's many X-linked recessive disorders which are very apparent in males who are the product of consanguineous partnerships. For example, G6PD deficiency and X-linked agammaglobulinemia and hemophilia A and B, all hematological diseases. Hemophilia A and B being very common in consanguineous populations, and even the Russian royal family before the Russian Revolution. And there's also many connective tissue disorders like the muscular dystrophies and Alport syndrome. And so the X chromosome has a lot of baggage on it still, as compared to the Y, which unfortunately only contains very few protein coding regions, many of which are actually just important for sperm development. But let's return back to the Y chromosome. What exactly exists within it from a molecular genetic standpoint, now that it had shed almost everything except its pseudoautosomal regions and its sex-determining region? Well, this reduced recombination with its X homologue, of course, helped preserve male-specific function, but there are three key characteristics that I think really explain what's going on with the Y chromosome. Number one, as we've emphasized a lot, is degeneration, because without that recombination with its pseudo-homologue, most of the functional genes on the Y chromosome were lost naturally due to mutations, deletions, genetic drift, which also really helps us understand how important recombination really is to chromosomal integrity. Number two, in order to retain some of this important stuff it's still got, like the sex-determining region, the Y chromosome undergoes something called intrachromosomal recombination. As the name suggests, this means that the Y chromosome has regions that recombine with itself in order to preserve itself, and that necessitates palindromic sequences. Palindromes are classically words that can be read the same forward and backwards, like race car or taco cat. And these palindromic sequences are naturally required in order for these regions to pair up during recombination and reinforce each other, exchange with each other so that they retain their faithfulness. However, this redundant gene amplification has many problems, as we'll explain in the next slide. And just as a small point, the pseudoautosomal regions, they are simply there to pair physically during meiosis with the X chromosome to ensure equal allocation of X and Y, even allocation during the process of cell division and creating gametes. And thirdly, because the X chromosome is so important functionally, males only have one copy of it. And so what we do as males is we have dosage compensation, which is where X chromosomes in males are more highly expressed and enriched. This is kind of the opposite as to females, where females classically have something called mosaicism, where their X chromosomes are sometimes turned off. This is very apparent in the fur coloring of the calico cats, as we are taught in biology classically. So this further brings us into the topic of the structural instability of the Y chromosome. Because the Y chromosome is so rich with these palindromic and ampliconic repeats, this, of course, is designed to facilitate the process of non-homologous or intrachromosomal allelic recombination. But because these repeats are so repetitive, they're paired together at indiscrete locations, they can be misaligned, there can be unequal dosing in terms of their son or daughter chromosomes. And so non-allelic recombination often results in things like structural changes, deletions, duplications, and inversions of the genome. In fact, researchers at the University of Chicago estimated that there are about 4 major inversion events that have happened in the evolution of the Y chromosome itself in the past 300 million years. Entire inversions of the chromosome, where the chromosome is literally turned upside down and placed back in an inverted position. So this gives you an idea of how structurally unstable the Y chromosome is, and that lends itself to many of the problems we have with the Y chromosome in terms of its phenotypic repercussions. So this brings us again to the topic of spermatogenesis. Well, spermatogenesis is a complicated topic. It happens in the testes of males. But the important part to consider here is that the recombination and crossing over happens in one of the second phases of it, the meiotic division phase. And this is, again, highly sensitive to copy number variation, because this is when the process of non-allelic or intrachromosomal recombination is occurring. And once this occurs, this can produce very dysgenic sperm. And so now we can start to understand why the chromosomal instability of the Y chromosome lends itself to things like azoospermia, or male factor infertility. One of the common causes of azoospermia are actually these deletions within the long arm, the Q arm of the Y chromosome, because the YQ arm contains 3 azoospermia factor regions, AZFA, AZFB, and AZFC. They cause a variety of infertility-like syndromes. They obviously cause azoospermia, or misdevelopment of the sperm, but they can also cause other structural changes within males, for example, short stature, because the Q arm actually contains short homeobox stature gene, and gonadoblastoma, which is a form of germ cell tumor. And so there's even structural changes like the ring chromosome, as you can see here, where the Y chromosome literally forms a ring, and that's, of course, not advantageous either. And all of these changes are due to this process of non-allelic or intrachromosomal recombination, because these repetitive sequences, they tend to involve or derive into things like duplications, inversions, deletions, aka just gene loss and copy number variation in general, which is not good for any complex chromosomal structure. And these AZF regions, you can see just for clarification, this is the long arm or the Q arm where the AZF regions are located. At either end, of course, are the pseudoautosomal regions. The AZF-A, for example, codes things like ubiquitinase, which may be important for certain protein development and structural formation. AZF-B is also similar, and AZF-C has a very high density of ampliconic or repetitive sequences. And this causes many enigmatic variants and many structural variants like isodicentric Y chromosomes, which can even lead to things like sex reversal within the severe range of this phenotype. Of course, this exists in a gradation. But this is just to give you an example of what the Y chromosome contains and how its structural instability lends itself to some of these drastic phenotypic consequences. So I hope you learned a lot from this video. Please remember to like, comment, and subscribe, and comment about what you want to see next in the future. Thank you.